smoking and I watched it and I was like,
Okay, then it looks like we're probably all here. Do you mind if I go ahead and get started?
yeah, I'll hold for in this room.
Maybe we'll get on 3 minutes.
Hard time right now.
Okay, cool. Then we'll get started. So we had some questions, and that's nice. It showed up, and then I put an answer as well, so you can have a look at that. So I answer them on there, but we'll go through them now, so you don't have to read that. I know no one likes to read. So let's take a look at the questions. The first one asks how astrocytes regulate blood flow. Can we draw this in a simpler form?
I have pictures, I'm sorry.
Me too. So no need to be sorry there. So we got options. As you might imagine, something as important as determining how much blood we move to different areas of the brain should probably have more than one way of working itself out there. All right. So we'll bring up this picture just so we have some idea of what's going on, and we'll try to simplify it just a little bit. The problem that we have to solve is, how do we know which parts of the brain are a little more active and thus need a little more blood? So we can listen to see when are neurons active. There's a couple things that you can look for with that. One would be neurotransmitters, and that's what this is showing us here. There's another option though, because not every synapse uses glutamate. And metabotropic GABA receptors aren't really going to get the job done here. So you can also listen for potassium. Now that's not showing up here, but astrocytes of course buffer potassium. All right, when they pick up that potassium, they can spit it out, and that potassium is going to then affect the size of the blood vessels. But it'll also stimulate this same pathway here. So GABA can get the job done. We just won't think about it, but just keep in mind that can work too. We're going to look at glutamate because it's a little more straightforward. And I suppose a glutamatergic synapse would be a little bit more expensive because it's going to be excitatory. And we're increasing the likelihood of action potentials, and those aren't free either. So I guess we're pretty good on neurons. If they make nitric oxide, this just diffuses freely through membranes. And nitric oxide is going to be a vasodilator. So it's going to affect the ability for myosin to bind to actin in smooth muscles. Smooth muscles are a little different. It's not so much about calcium, it's about post-translational modifications to myosin. So they have myosin light chain kinases and phosphatases that are going to determine whether they're combined. So smooth muscles are a little bit different than striding muscles, but it's the same idea. If they constrict, then they shorten their length, and we'll get vasoconstriction, decreased blood flow. If they relax, so if myosin doesn't interact as well with actin, they're going to die. Neurons are pretty straightforward, I suppose. The trickier one is going to be the astrocyte, and of course, these are going to have to be able to do both things. They're going to have to be able to cause an increase and a decrease in blood flow. Any responsible biological system is going to have the gas and the brake. Right? You can't just have one. It's not going to work. There are very, very few cases where we just have gas. Okay, didn't think about that, but yeah. So it could be in childbirth. That's a great example. You don't want the brake on that. You want that to go ahead and go to completion, cell cycle. You want that to go to completion. You don't want to stall there. Both of those don't end well if you stall them. But in most cases, we're going to have gas and brake. All right, so astrocytes have to do that because of their relationship with the blood vessels. So keep in mind right here, we're going to have those astrocyte input processes kind of surrounding this little area here. So they're wrapping around and giving the blood vessel a nice little hug there. And so these cells are going to be much better situated to communicate with the blood vessels than these cells. We keep neurons a little isolated from the blood. Blood's toxic to neurons. So we have that astrocyte middleman. All right, so how do we sense glutamate? How would we be able to know if neurons are releasing glutamate? What's the simplest solution? It's on this cartoon. Exactly. Put a glutamate receptor on the cell, so it can sense glutamate. It now has a receptor for that ligand. So whenever glutamate's released, it stimulates receptors on the astrocyte and kicks off some sort of intracellular signaling cascade. That's what we have here. So we'll learn about this a little bit later, but our NEUR411s are going to come in several different varieties, and this one is going to be GQ coupled. All right. Are we familiar with G proteins? All right, cool. We'll talk about those in lecture 9 in more detail whenever we get more into this. Now, this stuff matters because it's how we pay the bill, and it's how we are able to create those ionic conductances that this unit's all about. So we're touching on next unit. We're going to dive deeper into it once we get into it. So we're going to get an increase in intracellular calcium. All right, if you're a fan of simpler flow charts, the instigating event here is glutamate release. That's going to kick this whole thing off because that's our proxy for neuronal activity. We're active, so we need more blood. We can pull out glucose, oxygen, and make ATP. Anytime we're active, we need ATP. So that glutamate that's released is going to stimulate NEUR411s on astrocytes. Those NEUR411s are going to lead to an increase in intracellular calcium. All right, because they're GQ coupled. So at this point, we're in astrocytes. This calcium is going to do several things. One of the things that it can do is stimulate potassium channels. So we get potassium efflux out of the astrocyte. Of course, astrocytes are right next to blood vessels. Here's our blood vessel. We have some RBCs in there. These are, of course, made of cells. That potassium that exits as a result of that increase in calcium there is going to build up outside. Now, you might be thinking depolarization. And that would be true if we didn't adjust the permeability to potassium. So certainly the reversal for potassium is going to depolarize it a little bit. But the big thing here is that we get an increase in our permeability to potassium. So we move toward its reversal even further. Now these smooth muscle cells, if they're constricted, are going to be a little depolarized. They're going to be pretty far from potassium's reversal. So if we're going to be able to cause vasodilation, we're going to be a little depolarized. We're going to hyperpolarize them. And the way that we do that is by increasing potassium permeability. So quite surprisingly, whenever you increase extracellular potassium, it causes hyperpolarization. It's not because of a change in the reversal. That actually goes in the opposite direction. The reversal depolarizes. Look at the Nernst equation. That should make sense. We got to go to that Goldman-Hodgkin-Katz equation to see the real answer. Sure, we get a little bit of an increase in the reversal, but we get an increase in our permeability to potassium. So even though it's a little depolarized, it's still hyperpolarized relative to the cell. So we move toward its still hyperpolarized reversal. Whenever we get hyperpolarization, we get vasodilation. So the muscle cells relax. Here's one avenue. Because neurons were active, we get vasodilation. Now the other possibility is that these mGluRs are going to stimulate then phospholipase A2. And there's a few ways of doing that. It could be downstream of the calcium. Phospholipase could just be coupled to the G protein. Just to match this cartoon, we'll go ahead and put it downstream of calcium here. But it doesn't necessarily have to be. There's a few routes. Okay, but we activate phospholipase. This is an enzyme that's going to cut phospholipids. All right, so the name should make sense. There's a few different places that you can cut different phospholipids. So there's a few different phospholipases, and they all cut in their own unique place. Now, when you stimulate phospholipase A2, you're going to create arachidonic acid. So now we've made arachidonic acid, and this is a key regulator of blood flow. Now, it won't do anything, but its metabolites, or its downstream products, are the ones that are going to have either dilating or constricting effects on the blood vessel. So it really depends on how much, for example, cyclooxygenase activity that we have. So arachidonic acid can be metabolized by several different enzymes. And depending on which enzyme acts on arachidonic acid, that then determines the product. Just like which phospholipase is going to act on the phospholipids is going to determine the product. Are we making IP3 and diacylglycerol? Well, that's phospholipase C. But this isn't phospholipase C, so we make arachidonic acid instead. So we got a couple options here. And I'm not too familiar with arachidonic acid's metabolism and how the different enzymes are regulated. But what you'll notice is that we have the gas and the brake from arachidonic acid. So, either within the cell or by extracellular cyclooxygenases, we're going to make prostaglandins. Have you heard of prostaglandins before?
No.
Okay, good. So these are involved in inflammation. One of the things that they do is cause vasodilation. So they're going to do it in a slightly different way. They're going to act on prostaglandin receptors. These are GS coupled. They're going to stimulate protein kinase A, which then inhibits myosin light chain kinase. Same output though, vasodilation. Now I'm sure there's some negative feedback where prostaglandins are going to inhibit this pathway. This is just basic biology here. You see negative feedback all the time. And so whenever we have enough vasodilation, so after we get this vasodilation, arachidonic acid can escape without being broken down by cyclooxygenase and taken up into the vascular smooth muscle cells. Now they're going to process arachidonic acid a little differently, and they're going to make vasoconstrictors. So there's your gas and your brake. So there's a few different things that can happen as a result of arachidonic acid production. Most likely what we get is initial vasodilation followed by vasoconstriction because of negative feedback. We can bias ourselves toward vasodilation or constriction based on how acidic is our environment. Do we have enough oxygen around? If we have enough oxygen around, we don't need to bias ourselves toward vasodilation. So think of it as a balancing act based on your pH. What is it? Of course, we're going to be very, very close to neutral. We're not actually going to get in basic and acidic pHs. That'd be terrible, but we're going to move a little toward one or the other. All of that catabolic metabolism that we talked about, so breaking down glucose, this is going to decrease your pH. It's going to make it acidic. Has anyone ever cultured cells? Great. So usually you start off with a nice pink or red media, and then it becomes yellow over time. That's because of acidification. So those cells are living. That's a good thing. The fact that they're alive means that they're going to undergo metabolism. They're going to drop the pH, and that's what you're seeing. You're seeing a change in that pH indicator. So life is going to move us toward acidic pHs because we're always making ATP. This is going to drop the pH. You've probably heard of lactic acid, lactate, same thing. So this is what we're going to make with glycolysis, which is anaerobic. So if we're not able to break down that lactate further through aerobic metabolism, that's because we don't have enough oxygen around. When we use up our oxygen and create carbon dioxide, this is of course going to get absorbed in the blood, and that's what then affects the pH. We have our bicarbonate buffer system in the blood. So if we're very active, so when we're thinking acid, we should be thinking active. We probably need more vasodilation. And so the sensor that we're going to use for whether it's acidic, whether we're more active than our oxygen supply can handle, is going to be lactate. And that makes darn good sense because we start off with glucose. Anaerobic metabolism or glycolysis is going to create, we'll say, lactate. And then aerobic, which uses oxygen, is going to then break it down further into carbon dioxide and our 30 ATP or cell. If we don't have enough oxygen, lactate's of course going to build up because we can't drive this reaction. When lactate builds up, that tells us we need more oxygen, we probably need more vasodilation. So we have regulation of prostaglandin reuptake. Let's draw the border of our astrocyte there. So these prostaglandins can get taken back up into the cells. And this reuptake is going to be inhibited by lactate. So if we don't have enough oxygen around, we don't pick up quite as much prostaglandin. Those levels increase, we bias ourselves toward vasodilation. We can more rapidly eliminate prostaglandin signaling whenever we have sufficient oxygen. If there's enough oxygen around, we're breaking down our lactate. Lactate levels are kept low. Then we can pick up that extracellular prostaglandin, and we don't get quite as much vasodilation. So astrocytes are going to be spitting out a mix of arachidonic acid, prostaglandins. They're going to be picking up prostaglandins. Smooth muscle cells in the vasculature are going to be picking up arachidonic acid. If we pick up more prostaglandin, then the balance shifts towards arachidonic acid signaling. If we pick up a little less prostaglandin, well, then we're going to shift toward prostaglandin signaling. So it's all dependent on what's our metabolism looking like. Are we kind of hanging around here before the aerobic metabolism, suggesting we don't have enough oxygen? Well, let's cause a little vasodilation. If things seem fine and we're readily breaking down our lactate, no need for the vasodilation.
Wait, so when we're breaking down lactate, so when there's less lactate, there's less vasodilation that we just got at the end?
Yeah, exactly. Because metabolism seems fine. We're not stalling between anaerobic and aerobic metabolism, suggesting we have enough oxygen around. Yeah.
There's no need for this.
Exactly. Because the whole purpose of vasodilating is that we get more oxygen and glucose. Imagine that is legible. That's what's floating around in the blood that we care about. So we have to solve the problem of figuring out, do we need more oxygen and glucose? One way of looking at that is, are we able to carry this out? If we're able to do this, we probably are good on oxygen. All right. Is that simple enough? Yes?
Yeah.
Okay, good. Good. All right, then we had a question. So when I say energy substrates, think of that as the substrates of metabolism. So metabolism is a process, and it uses stuff. So the energy substrates would be the stuff. All right. Cooking is to metabolism what food or food is to substrates.
Yeah.
We get some food, we cook it. We get substrates, we metabolize it. And there's a few options. What's the principal source for energy in the brain? Right, glucose. Yeah. So there's one possible substrate that you could metabolize and create ATP. And there are other possibilities.
Nika's messaging me. I'm gonna check this.
She's here. Hold on.
Okay.
Maybe I'm muted. Dude, why is the whole fucking thing? Oh, okay. I can't unmute you in case you're trying to say something.
Oh, she didn't say.
Because this thing, I'm using HDMI. I'm sorry. Just text someone if you want to ask me a question. I'm sorry. Okay. But she's bright. She'll be fine. Okay. So why do postsynaptic potentials require more ATP than action potentials? Isn't that kind of surprising? Because in an action potential, we're going from -70 all the way up to +30, and then we're hyperpolarizing. It seems like we're moving a whole lot of ions, right? And we are. But you got to think about how many postsynaptic potentials do we have to generate before we kick off an action potential. And it's a bunch. It's a bunch because we're going to lose quite a bit of charge in the dendrites. We're going to have inhibitory input as well that we have to overcome. So it's not just you need enough to reach threshold. You need enough to reach threshold after taking into account all the inhibitory input that neurons are receiving. And so when you look at the dendritic arbor, this is of a single neuron, so not the forest, but the arbor, it's got quite the surface area. Lots of synapses are forming on the dendrites. So you just have a far greater number of postsynaptic potentials than you do action potentials. So it's just a numbers game there. There's just so many of them. Each individual postsynaptic potential is not going to be anywhere near as expensive as each individual action potential. But so many postsynaptic potentials go into making each action potential that they end up being more costly as a result. All right. So, any more questions? Just holler if they come up. We'll run through the quiz here. So the name section's still solid. All right, sodium, potassium, ATPase. I'm sure you could maybe roundabout say it has something to do with packaging neurotransmitters, but that's just a proton pump. It's doing that. It's not this. It's a different ATPase. Doesn't do any of this. Glucose. It's all of that and more. You got that. So astrocytes. Okay, so we're not going to transport glycogen to neurons. Glycogen is fairly large. It's going to be insoluble. It's not going to float around. We're going to break up glycogen into glucose, maybe even take it a bit further, break it down to pyruvate or lactate and shuttle that stuff over. Collectively, we'll call them energy substrates, whether it's glucose, pyruvate, don't matter. What's up?
It's been a minute since I did biochemistry. glycogen is when glucose is like stored.
Yeah, put a whole bunch of glucose together. Yes. So a big old polysaccharide, and it's insoluble just because it's so large. It's not just like floating around all over the place like glucose. And we don't want to give neurons glycogen. They don't know what to do with it. They don't make it. Thus, they don't break it down. But we are going to give them stuff. We have to give them stuff. Neurons aren't going to be connected to the blood supply directly. We got to give them things. They don't take care of themselves. Okay. So if we release arachidonic acid, it's either not going to do anything or be picked up and metabolized to 20 heat. I wouldn't think about that one. Nitric oxide is definitely going to increase cerebral blood flow. That's going to cause vasodilation. It's going to reduce the resistance within our blood vessels, and thus we get more flow. V equals IR is still true, even when we're not in neurons. Did you know that they figured out V equals IR outside of neurons? It's amazing, but some people don't study neurons. All right. Let's go through these then. If anything comes up, just let me know. We already got this. You told me glucose. Thank you. All right, so we got action potentials, we got synaptic potentials. Let's think about what's going on in these. We got sodium coming in, we got potassium leaving. There's probably that going on simultaneously during synaptic potentials as well. These are passive. So why do they cost energy? Why is it every time we're active, we now have metabolic demand? Yeah.
Because we have to regulate the flow of those ions. Like the neurons essentially taking advantage of how the ions naturally just sort of constitute move. So
yeah. Sodium's going to flow in. How do we make sure that sodium's always going to flow in passively? Not a gate.
Pump.
Thank you. Yeah, pumps, exactly. Yeah. So we gotta take it back out so that we keep that potential energy there. Yeah, exactly. So anytime you take advantage of something passive, you gotta restore it. You gotta create and maintain those ionic imbalances. So here we're clearly disrupting them. Sodium's coming in, but we have to keep higher concentrations out. Therefore, pumps must be activated, and those ain't free. Even free stuff isn't free. Okay. Besides glucose, what are some other options that we have? Great. Great. Fatty acids, ketone bodies. We could use those. When do we use those? Okay, great. Not great if you're starving, but yes. Yeah, when else might we be biased toward fatty acid use? Oh, what a nice life you live. You haven't had kids. You're not thinking about little infants that are drinking breast milk. They're awful, but that's their job. They're great because they're awful. They make you have to be better because they're so bad. But their diet's full of fat, and therefore, of course, they're using a lot of fat. So we all do this. We all, at some point, are using a whole lot of fat to keep the bills paid in the brain and to make the myelin that we're creating during myelination. Anything else that we might pick up out of the blood to pay the bill? Okay, still glucose, still a solid answer. Or it could be the lactate, pyruvate. Those are floating around in the blood too. Yeah, exactly. So it's the same story. It's just, are we picking it up from the beginning, or are we already in chapter 2 after glycolysis? Yeah, so we have some options. Now, of course, primarily we're going to be using glucose. And we're going to break down glucose through glycolysis, which is anaerobic, and then oxidative phosphorylation, which is aerobic. So in the cytoplasm, in the mitochondria. Okay. So let's fill out this table. This is essentially our starting, so our reactants. What are we starting with? And then breaking down into some product. And then we have a division of labor in the CNS. So certainly both astrocytes and neurons can carry out both of these, but it seems that there's a bit of disparity between them in terms of how much glycolysis are they doing relative to aerobic and how much aerobic relative to glycolysis. There's a bit of a division of labor. Of course, they're both still living cells that can do both of them. But it looks like one tends to do more glycolysis and the other tends to do more aerobic, and that should make a whole lot of sense. So let's start off with the substrates. Start at the beginning. So what are we starting with in glycolysis?
Glucose.
Thank you. We're going to start off with glucose. All right. What are we starting off with in aerobic metabolism? Excellent. Pyruvate, lactate, same thing. All right, so what are we making in glycolysis? Excellent. Hey, look at that. Nothing tricky here. We'll make a little bit of ATP. So this can pay the bills, but not well. What's our product in aerobic metabolism? Nice. Yeah, exactly. We're going to make some carbon dioxide. That'll diffuse out. And we're going to make a lot more ATP. So this would be if you are, let's say, an assistant professor, and this is if you're an MD. So a little bit of a different payout. Both doctors, but not really. Okay, so they both can pay the bills. One pays the bills a whole lot better than the other. Let's think about which cells are going to carry these out a little more so than the other. So astrocytes, are we thinking more aerobic or glycolysis? Yeah. Exactly. And then just look at how much you're getting. What's the payout? And who's more expensive? Yeah. Yeah, so those are the expensive cells. They got expensive tastes. They like to move ions a lot. So they're going to need more ATP as a result. Much humbler cells, the astrocyte there, no action potentials. All right, they're not changing their membrane potential. Oh, I'm positive now, and now I'm back to negative. They're not redecorating their membrane potential all the time, so they can get by on a little less. Okay. When I think about astrocytes, I like to think of them as mothers, and neurons are the little babies. I think I've said that before. So, mother has to feed baby. What are we going to send over to neurons to replenish their glutamate? Let's just think about the glutamate here. Because when you spit out glutamate, well, now it's gone. You got to remake it. Now, glutamate is a very important neurotransmitter. This is the excitatory neurotransmitter for our CNS. We need this. It's also just an amino acid we use to make proteins. Every cell needs this, neurons especially. So what are we going to send back to neurons so they can remake their glutamate? Yeah. Nice, glutamine. We'll probably have to send them some lactate at some point. But let's not get too far away from just glutamate. So we'll send them glutamine. Why are we sending them glutamine? Any ideas? Let's say we pick up glutamate. And then we just spit it right back out. There you go. Yeah, we probably don't want to just spit glutamate back out. Why did we pick it up in the first place? So we got to turn it into something else. Glutamine is a really nice option because it's a one-step reaction from glutamate to glutamine and glutamine to glutamate. Really nifty stuff. Glutamine synthetase is going to make glutamine from glutamate, and then glutaminase will break down glutamine back into glutamate. A nice one-step reaction. Glutamine synthetase will create glutamine. Have you ever heard of glutamine receptors? Have you ever heard of them at the synapse? I haven't. They're glutamine transporters, but I've never heard of a glutamine receptor. So we're not going to affect the activity of the neuron. We're just going to give it a substrate that in a single enzymatic reaction will replenish excitatory neurotransmitters, and we won't affect the activity of the cell. It's nice and quiet. So it's essentially inert as far as the membrane potential is concerned, other than the little bit that we use to bring it in. But it's certainly not an excitatory amino acid. No, no, no. What the heck is this? That thing on the bottom, that alpha KG. Nice, alpha ketoglutarate. Yeah, exactly. Why does the cell have that floating around in it? Haley, I haven't heard from you yet.
I'm sorry, what is it?
I haven't heard from you yet.
And then what were you asking?
What do we use alpha ketoglutarate for? When have you ever heard of that? Maybe you haven't.
Have it? Other than when I read it in the notes?
Okay, nice. Okay. So it's somewhere in the notes.
Yeah.
It's probably doing something.
Isn't that in the citric acid?
Yeah, there you go. So that thing that we have to do to create the reducing agents needed to drive oxidative phosphorylation, the TCA cycle, the citric acid cycle, whatever you want to call it. We're going to take our pyruvate and we're going to turn it into 3 carbon dioxides and then some reducing agents. So things that will carry electrons from the pyruvate to the electron transport chain and then drive oxidative phosphorylation. This is an important component of the metabolic machinery within the cell, but we're willing to sacrifice a little bit to keep our babies running. Parents make sacrifices, and astrocytes do the same thing. And luckily, they don't need nearly as much ATP as neurons, and they have those anaplerotic reactions that will regenerate alpha ketoglutarate. But this is another option that we have. We'll give over our very lifeblood, if you will, one of the things that we need to keep ourselves alive, we'll hand it on over to neurons. Have you ever heard of an alpha ketoglutarate receptor at the synapse? I wouldn't imagine, they don't exist. So this is another essentially inert substrate that we can hand on over. We don't have to worry about excitotoxicity. Neurons can pick it up and they can make glutamate, and we can keep this whole system running. Okay, so we got where we got those from. Okay. So whenever we have glutamate release, with this we're thinking neuronal activity. How is it that neuronal activity is going to then stimulate glycolysis in an astrocyte? We know we're active, we need ATP, so it would make sense that activity would stimulate glycolysis. Who can run me through that?
I write down that if you increase glutamate, you'll decrease ATP, so then you need to go through glycolysis to increase ATP.
Yeah, that's a great basic answer to it. So how does the glutamate release cause a decrease in ATP, let's say, over here in the astrocytes. What are they spending ATP on? Let me draw this a little bit differently. So we have glutamate release. That's what this is. There you go. Here's our astrocyte. We got a nucleus here. All right, so we've spit out glutamate. Now what? What does the astrocyte do to that glutamate? Excellent. We got to pick it up. And I always like to draw these as a funnel. So we have excitatory amino acid transporters that pick up excitatory amino acids, glutamate. Okay, so we got glutamate in the cell. Now what? Was that free? Okay, great. So this right there in that moment is free, but in the long run, no, it's of course not free. Because we're going to bias glutamate toward uptake by riding along some passive currents. What are we dealing with? Is this 2? I forget. I don't remember my stoichiometry very well. But we're letting sodium come in and potassium leave to bias us toward the influx of glutamate. It's very unlikely that this would reverse because we're using ion currents to drive that transport. The movement of these ions drives that change in shape needed to pull glucose in. This could run in reverse. It's just not as likely to happen because we have these ion movements that are biased toward their direction that drives influx of glucose. So we've let sodium leak out. We've let potassium leak in. How do we fix this? Thank you. The sodium-potassium pump. That's going to move sodium out and potassium in. Is that free? Exactly. There's your real cost here. So we're going to be spending ATP indirectly to bring that glutamate in because we have to maintain the ionic imbalance that's driving glutamate uptake. So sure, that glutamate transporter is free as long as you pay the bill downstream. So now we got glutamate inside. We'll turn it into glutamine and send it back. We already heard that story. Okay. The real key thing here is that drop in ATP. Well, now what? Yeah, you got to make more ATP. How does the cell know that it needs to make more ATP? Okay. Yeah, absolutely. So that's really important for excitable cells like beta islet cells and neurons. Let's not think about them here though, but those are really important. ATP sensitive potassium channels, no doubt. That is one of the ways that a cell can tell about how much ATP do I have and regulate its excitability accordingly. Phosphofructokinase is one of those enzymes involved in glycolysis. So taking glucose and turning it into pyruvate. I'm going to brush all the other reactions under the rug. This is inhibited by ATP. When ATP levels drop, inhibition of glycolysis drops. So we disinhibit glycolysis. We allow it to occur. So the rate of glycolysis is negatively regulated by ATP. If you like plots, think about it like this. Here's the amount of ATP we have, and here's the rate of glycolysis. If you have high levels of ATP, you don't need as much glycolysis. When you have low levels of ATP, you need to undergo more glycolysis so you can make that ATP. And so we do that by just having allosteric regulation of glycolytic enzymes by ATP. If we have a lot of ATP, it binds and it inhibits those enzymes. Only when we start to break down our ATP do we then disinhibit them and allow glycolysis to occur. Pretty straightforward stuff. It's a nice, simple system. So we know when a neuron's active because they spit out neurotransmitter. So we pick it up. That's not free. When we pick it up, we got to spend a little ATP to reestablish those ion gradients. When we break down ATP, we disinhibit glycolysis. Then we make more ATP. Okay. So neurons and astrocytes can both affect blood vessels. We've already gone through astrocytes. We kind of brushed aside neurons, but let's just go ahead and mention it. What can neurons do to affect the size of our blood vessels? Do they spit anything out that might? Yeah, exactly. Nitric oxide. That's a great option here. So nitric oxide is used by a variety of cells as a transmitter, as a way of communicating to their neighbors. It's brilliant. It doesn't get held up by membranes. It can just go wherever it wants, just diffuses freely. And it breaks down all by itself. So you don't need an enzyme to act as the brake. It has a built-in brake. You make it, it lives for a very brief period of time, and it goes wherever it wants. So it tells all the neighbors, I just made nitric oxide. And one of the things that it's going to do is inhibit smooth muscle cells. So it'll cause a little vasodilation. Astrocytes, well, we got some options. We can cause vasodilation. We can cause vasoconstriction. Right? That arachidonic acid is coming out and gets picked up. Well, we're going to metabolize it. If, on the other hand, we have higher levels of prostaglandins, we get vasodilation. So we got gas and brake. Really in both of these symptoms. I'm sorry, both of these systems. Nitric oxide has its own brake because it just spontaneously degrades. So you're not going to get prolonged vasodilation. Same thing is true for astrocytes. The arachidonic acid is going to create gas and it's going to create brake. The whole purpose of this is to scale how much blood we put into an area based on how active the neurons are, because that's an area that's going to need more ATP. If you're more active, you need more ATP. Thus, you need more blood, you need more oxygen and glucose. Just that simple. This is the basis then for fMRI. Only when you have a change in blood flow are you going to see a signal in your fMRI. So it's blood oxygenation level dependent. When you have an area of activity, that leads to vasodilation, so we get an increase in oxygen there. You might think we're going to see a decrease in oxygen because it's more active. No. What we're actually going to see is an increase. We're going to overwhelm that area with oxygen. So we'll actually see an increase in oxygenation in active areas. If there's an overt change in blood flow. Now, just keep in mind with fMRI, it doesn't actually tell you which neurons are active. Is it the excitatory neurons or is it the inhibitory neurons? That might matter. When you think about any brain area, there's going to be local circuit inhibitory neurons, and then there's going to be some projection neurons. They could be excitatory, they could be inhibitory. we'll make it excitatory. Both of these, if they're active, if we're firing action potentials, we're going to have to increase blood flow. But one of them decreases output of that brain region. Another one increases the output. So it doesn't actually tell you which neurons are active, and thus, is there a net increase in output or a decrease in output? All it tells you is that there's a change in blood flow. And you might not actually have a change in blood flow and have a change in the activity of an area. What if we're firing at 10 hertz here, 0 here, so we're not communicating, and then we have a shift. Now we're firing at 8 hertz here and 2 hertz here. There will be a big change in the output, no change in the ATP demand, and thus no change in blood flow. So with fMRI, you see where there's a change in blood flow, which suggests a change in activity, but a lack of a change in blood flow doesn't mean that there's no change in activity. You could have a change in the different cells, and we lack this resolution, but that's key. No output, output, polar opposite effects, but they show up the same in both. You'd have to, of course, look at their downstream targets and see if there's a change there. So you'd have to map out the entirety. Just looking at one little voxel and saying it's different means nothing. But it's our best option. Because, you know, people got to live. Can't kill them off in your experiment. So we need non-invasive imaging techniques. And the whole idea behind it is that activity and blood flow scale with one another. Unfortunately, the devil's in the details, and we don't have this detailed resolution with MR. Okay, so that's the unit. Any more questions? Yeah.
So our exam is Tuesday?
Yeah.
Will there be a quiz for it?
No.
Okay. You don't?
No.
Oh, right.
I wouldn't stand now.
No. So we won't have any class on Tuesday. I will send you an email on Tuesday that'll give you a link to a Google Doc. You'll make a copy of it, you'll fill it out, and you'll give it to me before class on Wednesday. I would try to do it Tuesday before the end of the day.
You can email it back.
Yeah, you'll just share it. There's a big share button. You'll click that, and you'll send it to jasonepit@gmail.com. It'll say it in the email. But yeah, that's it. You share it with me. It'll be great. You're probably wondering what the exam will look like. I'll send you a practice question. How's that?
Okay.
So you get some idea of what's this going to look like, or what kind of shit might I ask, at least for like one little topic. What I'm going to ask you to do. And so there'll probably be 3 questions I have you answer. Something like that. Anything else? Okay. Well, I hope you have a great rest of the day. And I won't see you on Tuesday. Bye, Minka.